Selection of primers, the oligonucleotide primers, is a key step for the success of PCR. Several steps are involved in selecting a primer which can be used for any PCR reaction. The first step comes the analysis of the target gene for which we are going to do amplification or designing a primer. We have to analyze the priming sites where we can prime the primer or attach the primer. Those sites should have some specific properties, like they should be free from homopolymeric tracts. There should not be repeated sequences of the same nucleotide. No tendency should be there for secondary structure formation, means the same primer should not have complementary nucleotides that can ultimately lead towards the formation of secondary structure, or the template DNA should not have any sequence that can make it secondary in structure. There should be no self-complementarity. There should be no self-complementary, no significant homology with other sequences of genome. That means every gene should have a specific sequence for which the primer can be designed. If the sequence overlaps or is common with the other pieces or parts of the DNA or genes, then non-specific amplification can be observed. Once the design of the primer is determined, we have to calculate the melting temperature of the primer, keeping in view the base composition of the primer. Out of many choices, we have to select well-matched primers that should have GC contents in between 40 to 60% and which can generate a product of appropriate size and base composition. It is also recommended that the amplified product should also not have the base composition more than 60%, the GC base composition. Once the primers are designed, keeping in view their GC contents and their ability to anneal with a specific fragment of the DNA, some refining can be done with the primers. For example, at 3' end, if we have G or C, it can give you good results. The two primers should not have complementary sequences, so that we should not get some primer dimers during amplification process. In any primer, no 3 consecutive nucleotides should be similar.